The connection between gut microbiome disruptions and obesity is no longer just a correlation, it increasingly appears to be causal. Studies over the past 2 decades have steadily built a case that the composition and function of the gut microbiota not only reflect metabolic health, but actively shape it. Changes in microbial diversity, the loss of key bacterial species, and disruptions in gut receptor function, particularly toll-like receptor 5, TLR5, can directly influence appetite regulation, insulin sensitivity, fat storage, and even behavioral tendencies around food. In short, an imbalanced microbiome can reprogram the body's metabolic trajectory. TLR5, a receptor that recognizes bacterial flagellin, offers one of the clearest windows into this mechanism. In healthy physiology, TLR5 serves as a gatekeeper. It detects the presence and movement of bacteria in the gut and modulates immune tolerance, intestinal barrier function, and microbial composition. But in TLR5 deficient mice, researchers have observed a remarkable phenotype. These animals eat significantly more, develop low-grade inflammation, become insulin-resistant, and gain excess weight. What's striking is that these effects emerge even without changes in caloric density or physical activity, implying that the loss of TLR5 creates an internal environment that actively favors metabolic dysregulation. Why does this happen? One theory is that the absence of TLR5 leads to unchecked growth of pro-inflammatory, flagellated bacteria, which in turn provoke immune responses and gut permeability, often referred to as leaky gut. This allows microbial products like lipopolysaccharides, LPS, to enter the bloodstream, triggering chronic low-grade inflammation that disrupts insulin signaling and promotes fat accumulation. At the same time, this microbial shift alters the production of short-chain fatty acids, SCFAs, and gut hormones like GLP-1 and PYY, which are central to satiety signaling. The result is a blunted fullness response and a stronger drive to eat, particularly foods that are high in fat or sugar. What makes this compelling is that when the altered microbiota from TLR5 deficient mice is transplanted into germ-free mice, the recipient animals also develop obesity and metabolic syndrome, even though they have normal genetics. This finding supports the idea that microbiota alone, shaped by receptor function or dysfunction, can transmit a metabolic phenotype. In humans, the picture is more complex but mirrors the animal data in key ways. People with obesity often have reduced microbial diversity, lower levels of SCFA-producing bacteria like Faecalibacterium prosnitziae, and increased levels of opportunistic, inflammation-promoting species. There is also evidence that certain microbiome configurations are associated with a reduced capacity to extract energy from fiber and a higher efficiency at extracting calories from otherwise indigestible carbohydrates. This may lead to a metabolic environment in which more calories are absorbed from the same quantity of food, again favoring weight gain. Additionally, disruptions in gut barrier function and receptor signaling can affect neural circuits that regulate appetite. Microbial metabolites and inflammatory cytokines interfere with vagus nerve signaling and may impair the responsiveness of neuropod cells, the gut's direct sensory interface with the brain. When these neuropods fail to signal satiety properly, the brain receives a distorted message that the gut is empty or unsatisfied, even after eating. This contributes to hyperphagia, persistent hunger, and overeating driven not by willpower failure, but by faulty signaling at the microbiota receptor interface. Moreover, certain diets accelerate this dysfunction. Diets high in saturated fat, refined sugar, and low in fiber promote microbial profiles that are less diverse and more inflammatory. They reduce the abundance of bacteria that produce butyrate and propionate, compounds that are key to maintaining barrier integrity, regulating appetite hormones, and preventing metabolic endotoxemia. The more these protective microbial signals decline, the more the system shifts toward a feed-forward loop: increased food intake, more inflammation, greater gut permeability, and deeper metabolic impairment. However, there is also good news. This system is not fixed. Targeted dietary changes, especially increasing prebiotic fibers, polyphenol-rich foods, and fermented products, can begin to reverse microbiome imbalances, support TLR5 function, and restore proper gut-brain signaling. Emerging research also points to postbiotics, metabolites produced by microbes, and symbiotics, combinations of probiotics and prebiotics, as tools to recalibrate the metabolic feedback loops at the root of diet-induced obesity. In conclusion, gut microbiome disruptions, especially the loss of key immune microbial sensors like TLR5, are not just associated with obesity, they appear to contribute directly to its development. They distort the body's ability to sense fullness, impair energy regulation, and provoke chronic inflammation that further derails metabolic control. These effects are not purely genetic or environmental, they are microbially mediated, which means they are also microbially modifiable. Understanding this causal relationship opens the door to new interventions that move beyond calorie counting and toward reprogramming the gut ecosystem to restore healthy appetite, metabolism, and long-term weight balance.